Good afternoon, everyone. It's a pleasure to see so many bright minds from our biology and pharmaceutical sciences programs today. For the next hour, we'll be embarking on a journey through one of the greatest triumphs of modern science, vaccine technology. This is not just a historical lecture. It's a comparative study of how our understanding of immunology and genetics has rapidly evolved. We're moving beyond the 20th century models, which relied on the pathogen itself, toward 21st century approaches that use genetic blueprints. Think of it as shifting from physical textbooks to digital code. The recent global challenges, particularly the COVID-19 pandemic, have brought vaccine science to the forefront, showcasing the power of new technologies like mRNA and viral vectors. For you, future scientists and drug developers, understanding the fundamental differences in these platforms, from live-attenuated to genetic vaccines, is absolutely critical. It defines the speed of development, the type of immune response, and the logistics of global distribution, especially in a vast and diverse country like India. So let's start with the basic principle before we dive into the technological evolution. Before we delve into the technical details, let's quickly frame our discussion. By the end of this lecture, you should be able to look at a vaccine, whether it's the MMR given to children or a modern mRNA booster, and immediately understand its mechanism and its advantages. We will specifically compare the three main traditional types, live-attenuated, inactivated, and protein subunits, with the newer genetic platforms like mRNA and viral vectors. Now, why does this matter? Simply put, vaccines are a cornerstone of public health. They are arguably the most cost-effective health intervention ever invented. Consider the eradication of smallpox or the near elimination of polio. These are monumental successes driven by vaccine science. The fundamental principle, regardless of the technology, is to introduce an antigen, a harmless molecular signature of the pathogen, to the body. This introduction trains your adaptive immune system. Think of it as showing your immune system a mugshot of the criminal. When the real pathogen attacks, the immune system already has the B cells primed to generate vast quantities of antibodies and the T cells ready to execute infected cells. All vaccine design, whether old or new, boils down to finding the most effective and safest way to present that mugshot to the body. Vaccine technology can be logically grouped into two major eras, which we refer to as traditional and modern platforms. The traditional or first generation vaccines relied on using the actual organism, either the whole bacteria or virus or a large, purified part of it. The challenge here is always managing safety. How do you ensure that the preparation stimulates immunity without causing disease? This approach often involves large-scale biological cultivation and chemical inactivation, processes that are inherently slower and less flexible. In contrast, the modern or second/third generation vaccines represent an engineering approach. Instead of growing the pathogen, we use synthetic biology and genetic engineering to deliver only the instructions for the antigen. The goal shifts from injecting the antigen to having the host cell manufacture the antigen itself. This is a crucial distinction. It transforms the manufacturing process from a slow, bioharvesting operation into a faster, more adaptable synthetic process. For instance, the newest category, nucleic acid vaccines, that's mRNA and DNA, doesn't inject any part of the virus at all. They only deliver the code. As we move through the rest of the lecture, keep this simple classification in mind. Traditional equals the pathogen or its product. Modern equals the genetic blueprint. Let's begin with the classic method, the live-attenuated vaccine or LAV. This is arguably the most potent form of vaccination. The mechanism is brilliant yet simple. We take the virulent pathogen and intentionally weaken it, a process called attenuation. This is usually done by growing it repeatedly in non-human conditions until it accumulates mutations that make it harmless to humans. The vaccine is live, meaning the organism can still replicate inside the host. Crucially, it replicates just enough to mimic a mild, natural infection, but not enough to cause serious disease in a healthy individual. Why is this so effective? Because the immune system sees the pathogen in its most natural form, actively infecting cells. This leads to an incredibly strong, comprehensive, and most often lifelong immunity. It activates both the humoral response, generating neutralizing antibodies, and the highly critical cellular, T-cell response. Think of the T-cell response as the search and destroy unit, essential for clearing out virally infected cells. However, the major trade-off is safety and stability. Since it's a live organism, there is a remote risk of reversion to virulence, though this is exceptionally rare with modern strains. More importantly, it is contraindicated for immunocompromised patients. Those with suppressed immune systems cannot safely handle even the weakened strain. Finally, live viruses are fragile, requiring a strict cold chain for storage, which presents logistical challenges in warm climates like ours. Moving to the second traditional platform, we have inactivated vaccines. Here, the approach is different. We take the whole virulent pathogen and kill it completely. We inactivate it, typically using heat or chemicals like formalin. This means the organism is structurally intact, but biologically dead. It cannot replicate under any circumstances. This offers a major advantage in terms of safety. Since there is zero chance of replication or disease, it is a much safer option for immunocompromised people and is stable enough to be stored outside of the intense cold chain required for LAVs. However, there is a trade-off in efficacy and potency. A dead pathogen doesn't stimulate the immune system as aggressively as a live one. The response is generally weaker and focused primarily on humoral immunity, antibody production. Because the pathogen is killed, it is usually processed outside of the cell, which bypasses the pathways needed to generate a strong cytotoxic T-cell response. To compensate for this weaker response, inactivated vaccines always require two things. First, multiple doses and booster shots to build and sustain protective antibody levels, and second, the inclusion of an adjuvant. Adjuvants, like aluminum salts, act as an irritant or a danger signal to intentionally ramp up the local immune reaction to the vaccine. Now we shift into the modern era with protein subunit vaccines. This platform refines the traditional approach by adopting a purely recombinant DNA technology perspective. Instead of using the whole pathogen, we identify the specific, most immunogenic component, say the hepatitis B surface antigen or the SARS-CoV-2 spike protein, and only use that. We don't get this protein from the pathogen itself. Instead, we insert the genetic code for this target protein into a production system, like yeast or insect cells, turning them into tiny, safe protein factories. We then harvest, purify, and package this single, highly concentrated protein. The massive benefit here is ultimate safety. By injecting only an isolated, purified protein, there is zero risk of introducing an infection, a whole virus, or genetic material that could potentially be misread. This makes them ideal for vulnerable populations. However, like inactivated vaccines, purified proteins are lonely antigens. They don't signal danger very well, so they're dependent on adjuvants to properly activate the immune system. The other challenge is the manufacturing complexity. Scaling up the purification of a stable, three-dimensional protein to billions of doses is a significant bioengineering task, which is why they are not always the fastest option for a rapid pandemic response. The viral vector vaccine is the first of our two genetic platforms. Here, we use a concept from molecular biology, employing a benign or disabled virus, typically an adenovirus, as a delivery vehicle or a Trojan horse, to carry genetic instructions into your cells. What does the vector carry? It carries the DNA blueprint for the target antigen like the SARS-CoV-2 spike protein. When you receive the injection, the viral vector enters your host cell. It releases its DNA into the nucleus, and your own cellular machinery then reads that DNA, creates the necessary mRNA, and produces the antigen protein. The vector virus itself is replication deficient, so it cannot cause an infection. This platform is powerful because having the antigen produced inside your own cell and then displayed on the cell surface is a very strong signal to the immune system. This leads to excellent activation of both humoral antibodies and, critically, the cellular T-cell response, mimicking the quality of immunity seen with live-attenuated vaccines. The main technical challenge lies in pre-existing vector immunity. If a person has already been exposed to the adenovirus used as the vector, their immune system may neutralize the Trojan horse before it can deliver its payload, potentially reducing the vaccine's effectiveness. This is a key design consideration for manufacturers like AstraZeneca and Johnson & Johnson. Finally, we arrive at the platform that has truly revolutionized vaccinology, the mRNA vaccine exemplified by Pfizer/BioNTech and Moderna. This is the ultimate expression of the vaccine as code concept. The key component is messenger RNA, or mRNA, the transient molecular script that your cells use every moment of every day to build proteins. The vaccine simply contains the mRNA instructions for a single target antigen. This mRNA is incredibly fragile, so it is wrapped in a protective shell of fat, a lipid nanoparticle LNP, to facilitate entry into the host cell. Once injected, the LNP delivers the mRNA into the cell's cytoplasm. I want to be crystal clear on a common misconception. The mRNA never enters the nucleus where your DNA is stored. It cannot, and does not, alter your genetic code. Your cell's ribosomes immediately translate the mRNA into the target protein. The protein is displayed to the immune system, and the mRNA itself is broken down and eliminated within days, just like your natural mRNA. The advantages are revolutionary. Speed and scalability are unparalleled. You only need the genetic sequence of the pathogen to start manufacturing. No live cell culture or bioharvesting is required. This plug-and-play design allowed for unprecedented rapid development during the pandemic. The highly localized and transient production of the antigen also leads to a powerful, balanced immune response. The primary historical challenge has been ultra-cold chain storage, necessary to prevent the fragile mRNA from degrading, though formulation science is rapidly improving this. This table summarizes everything we've discussed and provides a quick reference for your exams. If you understand the flow of this table, you've grasped the core of modern vaccinology. Notice the fundamental division down the second column: antigen production. Traditional and protein subunit vaccines use antigens that were produced externally in the lab. The viral vector and mRNA vaccines use the antigen produced internally by your own host cells. This is what drives the quality of the T-cell response. Look at the categories of safety/reversion risk. The Live-Attenuated platform is unique in retaining that remote risk, which is why it's not given to certain patient groups. All the modern platforms are inherently safe in this regard. Finally, compare the speed and logistics. The slower, more complex biological processes of the traditional methods contrast sharply with the fast, synthetic, and easily adaptable nature of the mRNA platform. This single factor has changed the timeline for future pandemic preparedness from years to months. This comparison is not about declaring one technology better than the others. It's about matching the right technology to the pathogen and the public health objective. In conclusion, where does the future of vaccine technology lie? It is not a winner-takes-all scenario. Traditional vaccines will maintain their relevance. When we need lifelong, durable immunity, as seen with diseases like measles, the superior cellular response of LAVs may remain the preferred option. They are reliable, and their logistics are well established. However, the modern platforms, especially mRNA, are the engine of innovation. The flexibility and speed demonstrated during the COVID-19 pandemic have unlocked potential far beyond infectious disease. Researchers are now extensively developing mRNA vaccines for cancer immunotherapy, aiming to program a patient's immune system to attack tumor-specific antigens. This is an exciting frontier. The main takeaway for all of you, as future biologists and pharmacists, is that vaccine design is now a multi-platform challenge. Our focus in India and globally is on ensuring we have a diversified and agile vaccine ecosystem that can rapidly respond to both endemic diseases and emerging threats, leveraging the proven durability of traditional methods alongside the unprecedented speed and precision of genetic technologies. I hope this has provided a clear, foundational understanding of the state of the art in this critical field. Thank you all for your attention and engagement in this complex but vital topic. Remember, vaccines are a testament to how molecular science directly translates into life-saving public health policy. I am now happy to open the floor for questions. If you have any queries about the mechanisms, the comparative pros and cons, or the future directions of any of these platforms, please ask in the comment section. I will see you in the next lecture.
Ciao.